Good evening to all the EAPCET aspirants. So now here we are with the chapter virus. So previously we have dealt with bacteria from microbiology, and now this is the chapter from virus, after which we will solve the EAPCET PYQs on total both bacteria and virus. Now let's get started with what are the concepts that we are going to look from virus. First, the basic introduction of virus, discovery of viruses, different viral particles, classification of virus based on their shapes, and after which we will be looking into the taxonomy of viruses and nomenclature of viruses, and then harmful viruses, and at last the reproduction including life cycle. Both lytic and lysogenic cycle will be learning out. So now if you see the basic introduction of viruses, firstly, viruses have few synonyms, like they are also known as biological entities. They are addressed as biological entities, biological entities or infectious agents. Okay, they are addressed as biological entities or infectious agents, but not a living cell, because a living cell has the basic characteristic phenomenon where a single cell should contain cytoplasm with a cell membrane and a nucleus. If a cell has cytoplasm, nucleus and cell membrane together, then it is addressed as a living cell. Okay, where virus has few synonyms, it can be addressed as biological entities or infectious particles or agents. Now, each virus will have a nucleic acid. This nucleic acid can either be DNA or RNA, but not both. Remember the statement clearly. It can have either DNA or RNA as the genetic material, and this nucleic acid is encoded by a protein coat. Okay, nucleic acid and a protein coat, that's it. Here if you can see, the single stranded DNA is encoded with a protein coat, that's it. Few of the virus particles or few of the infectious agents will also have envelope over the protein coat. Over the protein coat, they will also have the envelope. Envelope will be consisting of lipids, proteins and carbohydrates. Okay, the nucleic acid can be either DNA or RNA. It can be single stranded or double stranded. That will be encoded by a protein coat, but there is no point of cytoplasm inside the virus. So it is just infectious agents or protein particles, nucleoprotein particles we will address it as. Few of the virus will also have envelope over the protein coat. Virus are obligate intracellular parasites. Okay, they are the obligate intracellular parasites. They cannot reproduce unless they invade a specific host cell. Okay, unless they invade into a specific host cell, they cannot reproduce. Okay, they use host genetic and metabolic machinery. Okay, they use host genetic and metabolic machinery to produce the progeny. Okay, and they can cause serious damage and diseases in humans, plants and animals as well. The scientific study of viruses is addressed as virology. Okay, this is the basic introduction of virus morphology and study of viruses. And then here, viruses do not exhibit most of life processes of a cell. Okay, it doesn't exhibit any life processes of the cell. However, they maintain the genetic continuity. Okay, they maintain genetic continuity through multiplication. They undergo mutations by making them more just inert molecules. Discovery of viruses, whole lot of discovery is done, and you have to remember three main scientists. One is Ivanovsky and Bajering, M.W. Bajering, after which W.M. Stanley. These are the three scientists you have to remember when it comes to discovery of viruses. First, let us look into Grigory Ivanovsky in the year 1892. He is a Russian pathologist who studied tobacco mosaic disease. He used filtered sap of deceased tobacco leaves. Okay, sap of deceased tobacco leaves was taken Okay, how did he take that? A leaf which is infectious is grinded using mortar and pestle. Okay, it is grinded and the extract is taken into a test tube which is centrifuged, and this is poured up to bacterial filter paper, and it can easily, the infectious agents have passed out through bacterial filter paper, with which he has identified that the infectious agents are lesser in size when compared to bacteria. Look into the theory. He used a filtered sap of tobacco mosaic leaves, and this sap is passed across the filter paper, okay, where this is designed to trap bacteria. Okay, even after filtration, disease symptoms appeared in the healthy plant. He concluded that filterable agent caused the disease, and this filterable agent, as it passed through bacterial filter paper, the agents are of lesser size when compared to bacteria. What did Dmitry Ivanovsky identified from the diseased tobacco leaves? He has grinded the leaves and he has poured into a test tube and then centrifuged it, and the sap is poured into a filterable bacterial filter paper, and the filterable agents which are passed through bacterial filter paper have caused infections in a fresh plant. But his identification is the filterable agent has passed through bacterial filter paper and the agents are in less size of bacteria, but he didn't convey that the infectious agents are causing disease in the healthy plant. Again, that point is not conveyed. Of course, the experiment is done, but that point is not conveyed by Dmitry Ivanovsky. He just conveyed that infectious agents are passing through bacterial filter paper, and this filterable agent molecular weight is less when compared to bacteria. That is what been conveyed here. And then, yeah, here M.W. Bajering, he is the scientist who has done the same process. The filterable agent has caused infection to the healthy plant. He has noticed this point and he has given out the statement contagium vivum fluidum, which means the infectious agent has also caused infection in a healthy plant. He called the causative agent, that is contagious living fluid, that is contagium vivum fluidum. This is causing infection in a healthy plant again. Next, the scientist W.M. Stanley in the year 1935. Okay, W.M. Stanley, what did he do? He has crystallized the virus particles. He has purified the sap and showed that virus could be crystallized. The protein particles he has crystallized. Protein particles whichever are in the coat, protein coat, he has crystallized it and named the virus as tobacco mosaic virus. Okay, he has named it as tobacco mosaic virus. These are the three scientists to be highlighted. One is Dmitry Ivanovsky, second is M.W. Bajering, and third is W.M. Stanley. These are the three scientists to be remembered, and one more scientist here, he is Frankel Conrad. In the year 1956, confirmed that genetic material in TMV is RNA. Okay, he used advanced techniques like ultracentrifugation and X-ray crystallography and electron microscopy to study the viruses and their structures. Okay, who is the scientist who said that the tobacco mosaic virus is having RNA? It is Franklin Conrad, and he has discovered that RNA is the genetic material in TMV by conducting some advanced techniques, by using some advanced techniques like ultracentrifugation and X-ray crystallography. These are the objective points to be highlighted, and this is about the complete discovery of viruses. Clear till here, ma? Any doubt anywhere? Next is classification of virus, which is very important. Classification of virus is by the standard committee. A committee is fixed to classify and place the viruses into different groups, and the committee name is International Committee on Taxonomy of Viruses. It is ICTV. Quite a times the question is asked from the abbreviation, that is ICTV, International Committee on Taxonomy of Viruses. This governs the classification and naming of organism, both Classification and nomenclature of viruses is governed by this committee. It uses 3 levels of hierarchical systems. One is family, second is genus, and third is species. Family ends with viridae. Fine. Genus ends with virus. Remember. Species has some English descriptions like based on the disease it is causing. For example, poliovirus. Okay. So, family has the suffix viridae, genus has the suffix virus, and species with some description especially on the disease it is causing. For example, poliovirus. Next, based on the disease caused by the viruses. For example, virus causing AIDS is named accordingly. Fine, light. It belongs to the family Retroviridae. See, viridae is the suffix, and genus lentivirus, virus is the suffix here, and species as it is causing to human beings and it is due to deficiency of immune system. It is named as human immunodeficiency virus, HIV. Okay, human immunodeficiency virus. Fine, right? The name, the species name is given with the disease description. Clear with the classification and nomenclature? And then structure of viruses. First, look into the size range of virus. We have the range from 300 nanometers. The range varies from 300 nanometers to 20 nanometers. 300 nanometers, for example, TMV, which is used for viral discoveries, and 20 nanometers, for example, parvoviruses. What are parvoviruses? These viruses belongs to a family which have smaller size. Okay, they belong to a family which has smaller size and it has single stranded DNA as the genetic material. Fine, right? These are parvoviruses whose size is very small in the complete viral range. So, virus ranges from 300 nanometers to 20 nanometers size. 300 nanometers example will be TMV, 20 will be parvoviruses. Clear, right? And then this is comparable to small bacterial cells like mycoplasmas and ribosomes. Clear till here? It is comparable to small bacterial cells which are wall-less bacterial cells like mycoplasmas. Of course, ribosome you can consider it like an organelle. Okay, it is not a bacterial cell, it is an organelle. Fine, right? You can consider like mycoplasmas only. Morphological types, we have 4 morphological types. Helical, for example, tobacco mosaic virus. Polyhedral viruses, for example, adenovirus, it has a envelope also. Okay. Helical viruses, for example, TMV, it has a genetic material and a protein coat. Whereas polyhedral virus will have the genetic material, protein coat, which is encapsulated with the envelope as well. Spherical virus, for example, influenza virus. Okay, spherical virus, example, influenza virus. This also has membranous envelope. Next is complex virus, for example, Bacteriophage, which has tadpole-like structure. It is complex, it is addressed as complex virus because it has head, collar, tail and tail sheets at the end, tail fibers as well. As the structural complexity is more in comparison with the other viruses, this is addressed as complex viruses and the example is Bacteriophage. All the different structures of viruses with the examples are important. Next. Yeah, here is the complete description based on the morphology. Helical viruses are rod-shaped viruses. They are like long rods, and their structure can be rigid or flexible. Examples, if you consider rabies virus and tobacco mosaic virus. These are the examples. Rabies or tobacco mosaic virus. Polyhedral virus, they have many sized shapes. They have many different shapes. For example, herpes simplex virus and polioviruses. Next is enveloped virus. Okay, enveloped virus. Viruses with a capsid enclosed in an envelope completely. Virus with a protein coat over which the envelope is present. This is roughly spherical. Examples, if you see, it is influenza virus and HIV. Complex virus, which are complicated structures, especially those infecting bacteria. These are the complicated structures, especially they are infecting bacteria. Examples include bacteriophages, for example, T4 bacteriophage. Next, some viruses, like if you see measles virus, they will have spikes or glycoprotein projections that help in host cell attachment. These spikes are from protein coat or capsid. If that is the question, the answer will be these are from envelope. Okay, like the spike-like projections with glycoproteins will aid in easy attachment of virus over the host cell. They can attach easily over the host cell. Okay, these glycoprotein projections are from the envelope of the virus particle. The spikes are crucial for the virus by binding to the receptors of the host cells. Remember, okay, important. Basic components of virus, as we have discussed, the basic components of virus include the core, that is nucleic acid. It can be either DNA or RNA based on the host cell it is infecting. Okay, and the protein coat for sure. Genetic material is covered by or encapsulated by protein coat. Few of the virus particles will also have envelope over this protein coat. Now you see core. This contains nucleic acid. It contains nucleic acid. For example, genetic material. It can be either DNA or RNA. DNA or RNA. Capsid, it has protein coat over the genetic material surrounding the core. This provides protection to the cell. Capsomeres, you see, over the capsid coat, it has protein subunits. These protein subunits are known as capsomeres. Capsomeres are protein subunits. Capsid is made of repeating protein subunits called as capsomeres. The number and arrangement of capsomeres are virus specific. It is specific to the viruses. Clear till here? Core is the nucleic acid. Next is capsid. Capsid will have protein subunits. Those protein subunits you call it as capsomeres. Clear, right? Now, genetic material in viruses. As we have discussed, the core is what? Genetic material, which is nucleic acid. That depends on which host cell it is infecting. It can be double-stranded DNA, it can be single-stranded DNA, it can be double-stranded RNA. If the virus is affecting a plant cell, then mostly the genetic material is single-stranded RNA itself. If it is infecting an animal cell, then it will be double-stranded DNA. Bacteriophages, the virus particles which are infecting bacterial cell, will have double-stranded DNA. Some viruses like HIV, which cause AIDS, they have two identical RNA molecules called as genomic copies. Fine, right? Now the shape of nucleic acid. Shape of nucleic acid, it can be circular or linear. It can be circular or linear. Fine, right? Next, most viruses will have a single nucleic acid molecule. Most of them will have single nucleic acid molecules. Clear till here? Any doubts anywhere, ma? And then tobacco mosaic virus. The size of TMV is around 300 nanometers long. Okay, its size is around 300 nanometers long and its diameter, it is around 300 nanometers long and its diameter is around 18 nanometers in diameter. Okay, 300 nanometers long and 18 nanometers in diameter. Whose molecular weight, if you see molecular weight of this virus, it can be 39 into 10 to the power of 6 daltons. Okay, molecular weight of TMV is 39 into 10 to the power of 6 daltons. Capsid will have protein subunits which have around 2130 protein codes called as capsomeres. 2130 proteins called as capsomeres. They are arranged in the helical symmetry. Each protein subunit will have 158 amino acids. Remember that each protein subunit, they have 2130 protein subunits called as capsomeres, each of which each protein subunit will have 158 amino acids. Number is very important. This contains single stranded or spirally coiled RNA with around 6500 nucleotides. Now you look into the RNA. RNA will have how many nucleotides in the single stranded RNA? 6500 nucleotides. So if you in a single TMV, if you have to take the ratio between the nucleic acid, the nucleotides, the number of nucleotides in the RNA and the number of protein subunits in the capsid, that is capsomeres. Okay, the number of nucleotides in the single stranded RNA and the number of capsomeres in the capsid. If you have to take the ratio, first number of nucleotides which are around 2000 nucleotides, 6500 nucleotides is to how many capsomeres? 2130 capsomeres. The ratio will be around 3:1. The ratio is around 3:1. Number of nucleotides is to capsomeres. It is 3:1 ratio. Fine, right? Any doubts anywhere, ma'am? And then now bacteriophage, which is T4 bacteriophage. Look into the structural description. At the center or at the core, you have DNA which is double stranded DNA. Okay, DNA at the center is double stranded DNA. It has a protein coat over it which is capsid, which has head and then collar and then which has sheath, okay, followed by tail, base plate tail and then tail fibers. At the end of tail fibers, it has the enzyme which is known as lysozyme, okay, which is lysozyme. Lysozyme is the enzyme which is at the base, okay. This lysozyme will help in degrading the wall layer of bacteria. Bacterial cell wall composition, mostly it is peptidoglycan, right? That wall layer will be degraded by lysozyme which is at the end of tail fibers in the bacteriophage. This resembles like a tadpole structure where head contains double stranded circular DNA and then tail includes sheath, base plate, pins and tail fibers. Functionality, if you see, tail fibers help attachment to the host cell. This tail sheath will inject viral DNA into the host cell. This is about T4 bacteriophage. And after which, if you consider multiplication of bacteriophage. Multiplication of bacteriophage, see, virus cannot replicate or multiply in the absence of host cell. Virus multiplies or replicates only in the presence of host cell. As such, two cycles are encountered based on the survivor or death of host cell. If the host cell dies because of viral replication or multiplication, then it is addressed as Lytic cycle. If the host cell survives along with the virus particles, then it is known as Lysogenic cycle. Lytic cycle ends with the death of the host cell. Lysogenic cycle where virus remains in the host without killing it. Yeah. Just a second. Yeah. So here if you see, first step is attachment. First I will discuss about Lytic cycle and how from here Lysogenic cycle is diverted. Lytic cycle you see, ma'am. First bacteriophage will attach to the host cell. This is known as attachment. This bacteriophage attaches on the surface of the host cell. The wall layer is degraded by the lysozyme enzyme. Okay, once the wall layer is degraded, a small pore or opening is created and the second step is penetration, okay, wherein DNA, double stranded DNA from the virus is penetrated into the bacterial cell. Now third step is biosynthesis. How this biosynthesis happens? The genetic material of virus which is penetrated inside the bacterial cell will make use of host machinery and it starts replicating. Okay, the viral genetic material will make use of the host DNA or the host metabolic machinery, and it will synthesize the protein coats and the genetic material. This is biosynthetic phase. And 4 is maturation. Now different virions, different protein coats and genetic materials, they will unite and they will form into a one complete virion. As such, many virus particles are formed and they mature. New phase particles are assembled here in this 4th stage, that is maturation. And 5th stage, which is lysis, what happens is the newly formed virions or the matured virions will burst open the bacterial cell and it will cause the lysis or death of the bacterial cell. Thereby the bacteriophages will be released from the bacterial cell. What happens in the lysogenic cycle? The attachment step, penetration step goes same. Here after penetration, what happens is the viral genetic material gets integrated with the bacterial genetic material inside the lysogenic cycle. Once the viral genetic material gets integrated with the bacterial genetic material, the biosynthesis happens and the viral particle will replicate along with the bacterial genetic material like this. The replication happens along with the bacterial cell. So here this will not cause the death of the bacterial cell, whereas in lytic cycle the replication of bacterial cell will not happen, only virus particles will replicate, it will burst open the bacterial cell and it will release. Clear till here? Understood, right? Now we will see the crystal clear differences between lytic cycle and lysogenic cycle. See, as we have discussed here, you can clearly see absorption and penetration is same. At the 3rd step, instead of biosynthesis in lytic cycle, here you will have integration, where viral DNA is integrated along with the bacterial DNA, which causes prophase. This step is integration followed by replication, where viral particles will replicate along with the bacterial DNA, and then synthesis followed by assembly and then release. Okay, these are the sequential steps of lysogenic cycle, and this involves bacteriophages like lambda are involved in lysogenic cycle. Unlike lytic cycle, there is no lysis or death of the host cells. Clear till here? Any doubts anywhere, ma? Now we will see clear differentiation between lytic and lysogenic cycle. Just a second, let the slide get loaded. Yeah. Yeah, here we go. First, lytic cycle will cause the death of the host cell, whereas lysogenic cycle where viral DNA is integrated with the host DNA and where the cell is dormant. Virus type is virulent type here. Here these are known as temperate phases. Integration, viral DNA does not integrate with host DNA. Here they will integrate with host DNA. Effect of host cell, here host cell is destroyed, whereas here host cell is alive. Latency, there is no latent phase, but here latent phase remains inactive. Viral DNA replication is independent here. Here this replicates along with the host DNA. Next, symptoms in the host. Rapid onset of symptoms are seen. Here lysogenic cycle, it is delayed symptoms. Examples T4 bacteriophage, here lambda phase, and here these are not needed for immediate lytic activation, and here stress, UV light and chemicals may trigger lytic activation in the lysogenic cycle. Here they will burst open, it releases 50 to 200 new virions. Here there is no virion release at all because virus replicates along with the bacterial DNA. These are the clear differences from lytic and lysogenic cycle. So I hope the complete virus chapter is clear to you. If we have to take the key notes of today's session, we have learned about viral discoveries, introduction and discoveries, followed by we have clearly dealt with the classification and nomenclature of viruses by taking a standard example of HIV, then a wide range of sizes starting from 300 nanometers tobacco mosaic virus to 20 nanometers parvovirus is the size range, and different shapes of viruses we have helical, polyhedral, icosahedral and complex viruses, followed by the components of virus, nucleic acid, based on the infection what type of nucleic acid will be, either DNA or RNA, single stranded or double stranded we have learned, followed by protein coat which has subunits called as capsomere. Few of them will also have envelope. The virus like measles will also have glycoprotein projections over envelope which will easily get attached with the receptors over the host cell, followed by we have learnt about two standard examples, TMV and bacteriophage in detail, followed by cycles, lytic lysogenic cycles, at last comparative studies. The concept is dealt on whole. In the next class, be prepared with bacteria and viruses together. We will be exercising EAPCET PYQs. Yes.